Peter Godfrey Smith said the exact same. He has this dilemma at the moment where he doesn't want to work with animals because he has such an idea of their consciousness from an ethical and moral perspective. He doesn't want to work on animal models. But then he doesn't believe computational models will work because it's sort of extracting, just one bit of information out of this crucial, really complicated, complex system. So it's a bit of a dilemma how we deal with that going forward.
Well, so what some people will say is what we need are brain organoids.
Yes, go to this. This is exactly, go to this. Great one. Great pivot. I have this down here. Great pivot. Go into organoids.
Okay, so these are structures that can be grown from human stem cells or stem-like cells. I'll say a bit more about that in a moment. But basically, if you grow them into neurons in a petri dish, well, in a particular gel, they have to be sort of three-dimensional, so they don't just spread over the surface. Then they grow as cells do, and they multiply. But also, because they are neurons, they have, in a sense, you can think of them as kind of programmed to assemble themselves into a brain-like structure, as they do in a growing embryo.
Right.
They don't have the surrounding tissues of the embryo that are giving them signals to sort of guide them. So, for example, to say, right, you need to grow something here that's going to become the brain stem. They don't have those signals. So they don't, this growing mass won't look just like an embryoid or a fetal brain, but it can look surprisingly, spookily like it in some cases. It also doesn't have, at the moment, vascular tissues, doesn't have a blood supply, so it can only grow so big before the cells inside are starved of oxygen. But there are ways of being able to give it a vascular system so that, in principle, we might be able to make these things bigger. So they are brain-like. And these are cells that are signaling to each other. They work. They're doing the same stuff as our brain cells are, and they differentiate as they do in a growing fetus. So you get the different types of neuron. You get structured tissue. And so they have been called, misleadingly really, but understandably, they have been called mini brains, and some of them do look a bit like that. So they're grown as ways of studying brain function and brain development, in humans. So these aren't, these are human cells. And I say all of this because I have had one grown myself from my own tissue. So I had a piece of skin, basically, taken from my shoulder. And so it's just like a normal biopsy. It was transformed into a stem cell-like state, and then those cells were grown into brain organoids. So I had several of these little things grown, and I've got a lovely sort of stained micrograph of them, when you can see the structure within them. So they could be of fantastic value for medical research. For example, the people who grew mine were actually growing brain organoids from people who had a congenital predisposition to early onset Alzheimer's, so a genetic condition that made that likely to happen. And so by growing their brain organoids, these researchers hope to be able to understand what it might be that sort of goes wrong that leads to Alzheimer's.
That's so cool.
And they've been used to study Zika virus and all sorts. But in order to do that, you get better and better information if you can grow a better and better mimic of real brains and make them sort of bigger and bigger and more brain-like. But the more you're able to do that, the more you have to start thinking, at some stage, do these things have some sort of consciousness in them? And then what? Then what responsibilities do we have? And no one really knows. But it does seem to be, I would think it is a genuine question. We're probably not at that stage yet, but we're getting better and better at growing them. So, that is, potentially, it's a system where we could really start to sort of answer some of these or explore some of these questions about consciousness and when it arises and so on. But it also comes with a host of ethical dilemmas.
Yeah, absolutely. It could be a great monetary opportunity for a business, grow your own organoids at home kit. Do you see an actual world where you could buy a kit and you're just taking your own skin cells. You have your three little chemicals that are inducing the cell to differentiate in the way that you want, and people are just growing their own brains at home. Do you think that could be possible? I mean, without regulation, that's definitely possible, right?
Without regulation, I see no reason why it wouldn't be possible. I
Yeah,
mean, I don't want to, by any means belittle the skill, the kind of craft
yeah,
that's needed by the people, who do this stuff. It's not simple stuff. It's not like growing, mold from some of your skin cells or something. But
yeah.
it's, but in principle, there's nothing that complicated about it if you have the right kit and the right sterilization procedures and all the rest of it. So yeah, it could be done without regulation.
It's so strange. And how do you think about the ethics of that? Like, it's just such a weird question. But if it's something that we're going to have to face probably quite soon, like how are you able to grow conscious agents in a dish? And if so, do those have rights? How should we think about them legally, ethically, morally, scientifically? A whole bunch of questions that we just have absolutely no idea how to answer.
We don't have any framework for thinking about this. I mean, when I had these brain organoids of mine grown, people then asked, how did you feel when they were killed and sliced up for the microscope? And I didn't feel like I had any duty to them or that there was anything wrong about that. But all the same, it wasn't like you know, a piece of my skin that I was, totally indifferent about. There was something, it was me. It was a bit of, it wasn't me. It was a bit of me, with my DNA that was growing, and that is, in fact, some of these cells are still, as far as I know, in the freezers at UCL. Legally, it wasn't me. This was interesting that I hadn't realized, and I wasn't worried about, but it was still quite striking. But once they've undergone a round of multiplication, of proliferation, they are no longer classed as, I have no legal right to them.
Interesting.
They're lab cultures. But how we think about it ethically, we don't really know. We don't know what it's like. We don't even know how to think about what that experience might be like, if it exists at all, of
Yeah.
a brain organoid. They have, for example, there are no pain receptors in the brain, so I'm told. So it's not going to feel literal pain. Some people say, would it feel existential pain? I mean, what does that even, how do we think about that? It has no sensory input.
Yeah.
So, it's the classic sort of philosophical brain in a vat question, but it really is like a little brain in a vat. But we might be able to give it sensory input. In fact, there are these pictures, you may have seen them, of some brain organoids that are grown. Our visual system, and our optic nerve is, in a sense, just an outgrowth of the brain. And there are some brain organoids that have been grown with these light sensitive patches on them, and they look like two little black eyes on these things. I mean, that is, in a sense, it's kind of what they are.
It's kind of
Light sensitive.
terrifying, right?
And I mean, well, yeah, how do we think about that? And, people have done this. They've connected brain organoids up to a piece of lab grown muscle so that signals from the brain can make the muscle twitch. So we can potentially give them some sort of actuation. So, it's certainly within the realm of possibility to give them access, if you like, to information around them that will feed in. So, what these questions mean for the responsibilities we have. Some people have suggested that we might get soon to a stage where brain organoids have a kind of legal guardian
Right.
that thinks about the rights, that we owe those systems. But does that make sense? So all of these are questions that and partly they're questions that we find hard to answer, because it's coming back to this question of consciousness. We don't have ways of deciding whether something is conscious or not.
Yeah.
So we have no real way of knowing whether it's meaningful to think about experience in these brain organoids. But we're going to have to grapple with questions like that
For
quite soon, now, really.
Yeah, sure. I think it's really interesting that they already have some legal definitions on it going through one one cycle of cellular replication, even though that's still 100% Philip Ball DNA, right? Like the cellular differentiation doesn't change the fact that that's still your DNA and genetics in those cells, but it's not legally yours. So that was kind of interesting. I think I would have been very or more dismissive of this before I was introduced to Mike Levin's work. You know, the stuff that he was telling me about, like they're trying to communicate with the Xenobots now by like, because they have some vibrational sensations. So they're trying to like vibrate the petri dish in a way, and they're trying to see if it can learn from that. Like, have you seen Black Mirror? Like, it's just a lot of these scenarios are just extremely Black Mirror like.
Oh, they've got to have a Xenobot episode of Black Mirror in the next season.
100%.
Yeah.
Let's hope it becomes a Black Mirror episode before it becomes reality, right?
The Giant Shoulder mission is to explore radical ideas in biology, neuroscience, and consciousness, and elevate those stories to the highest possible level while keeping them accessible to everyone. If this interests you and you want to support independent science, then please consider subscribing to the Clips channel. Check out our 26 Neuroscience book. You can download it for free